What are we at? 15. Okay. Those of you that are wondering what we're up to today, if you go to Classroom and go to the classwork section, this is what we'll be looking at. So the light reactions Google slide, you want that. And these two handouts that I'll be talking about. And then we've got a couple of videos we can look at later. So this is what you want for today. Let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. It looks like more people are missing today than yesterday. Check my email. Did anyone let me know about Eid? New. Okay. Yeah. So I'm thinking of an idea for me for next year because I think I am. I'm going to be teaching at the OCV again next year. You guys know the TV show Masked Singer? Well, I'm thinking maybe I'll do something like that. I'll be a masked teacher, not a medical mask, but I'll wear a Halloween mask, all quad, my first quad. See if anyone can figure out who I am. Oh, I thought of doing it this quad, but I couldn't find an appropriate mask. Most of my masks are a little grotesque, so. Okay, while we wait for a few million people to show up, here's a guessing game for you. What two genres of music do you think I follow? I do like it, but I'm not a huge country fan. Oh no, not into hip hop at all. Alternate? No, I can't stand Live 88.5. Hey, Grant's got one. Definitely metal. I'm a metalhead. I have been for years. I play classical because of my musical training, but I don't like, I don't go out and actively listen for it. Big in the 70s. Yeah. I really like is a mashup between disco and rock and heavy metal, or at least hard rock. Right? Like, have you guys seen the AC/DC Bee Gees mashup? Okay, it's bizarre. It is really bizarre.
I needed an online store for my jewelry design business, so Shop Here, powered by Google.
I admit the pants are a little disturbing. anyway, surprisingly, that works. But Metallica, yeah. Iron Maiden, Judas Priest, some Sabbath. I was also into punk while I was younger. Well, I call Aerosmith hard rock rather than heavy metal, but yeah, I like them. Uh, Probably my favorite punk bands were the Dead Kennedys, and the Smiths. The Clash were all right. Now, I'm guessing your genres probably run more rap hip hop than anything else for you guys, which seems to be the going trend. Listening to it's either that or alternate. Listening to stuff like what is that station? Uh, Not live. Yeah, live is alternate. Okay. All right. For those of you that weren't here a little bit earlier, if you want to pick up the slideshow, it's on uh, Google Classroom. This one here. It's called the light reactions. That's what I'm going to open up now, and we'll start taking a look at it in a minute. The other thing that you want to grab are these two. Okay. So I'm going to probably end up doing this diagram once it shows up as a Jamboard, right? And I'll redraw this out. But at least you've got a hard copy of it as well, if you want it. And then when we get to the end, the very last thing is how this works. This is also the very last slide on our PowerPoint or slide deck. And so this would be a good version to have, this or the version in the slideshow, whichever one you prefer. Okay. But where I'm going right now is this. So let me present that. If you haven't done the attendance thing, could you go off and do that? I'll wait a couple minutes. Those of you that are still here, let's see how good you are. In that picture of these plants, how many different kinds of plants can you identify? So obviously, that's probably a conifer, right? Because you can see the needly branches. Anyone remember what phyla the ferns belong to? This would have been all last year, grade 11. Then you can see the mosses here, right? The non-vascular, no, that pteridophyta, close. And then the mosses, the bryophytes, conifers. This is probably BC. Looking at it, because this is definitely a temperate rainforest, not a tropical one. Okay, let's get going and talk about plant and plant biochemistry. So what we're dealing with, right, is photosynthesis. So life from light and air. And by light, sunlight, and air, obviously oxygen. So energy needs of life. Okay, all life needs a constant input of energy. Why? Why do we always have to have energy coming in? Yeah, another way of saying that, is that life is endergonic. Okay, all the chemical reactions for growth are basically synthesis reactions, which are all endergonic. Okay, that's why we need a constant input of energy. Okay, so we know what heterotrophs do. We just looked at it under cellular respiration, right? We get our energy from eating others. You can be a herbivore, carnivore, omnivore, it doesn't matter. We have to eat something else. There are a few exceptions. And I'll talk about those exceptions in a minute. Okay, so we eat other food, which is basically other organisms, and what we want are their organic molecules. Okay, and then we make our energy, basically ATP, by respiration. Okay, yes, mushrooms are heterotrophs too because they do not photosynthesize. They're not green. Right. They're eating dead matter, but they are still eating others. Okay, what can you think of an animal that is an autotroph? Not a jellyfish. Single-celled organisms, cyanobacteria are not animals. No, those weird sponge things. You're closer. They're not. They're not green though. No, it's actually a sea slug. Okay, and they look like this. These are photosynthetic sea slugs. Okay, they literally take sunlight and use photosynthesis to make glucose. So they're doing photosynthesis. The question is, they're animals. How the heck does this particular group, and there's a wide variety of different morphologies or shapes in these guys, how in the world do these guys get chloroplast? Yeah, they have chloroplast. The question is how? Because their chlorophyll is in their chloroplast. Yeah, they eat algae, and then when they break the algae down, they confiscate the chloroplast from the algae and incorporate it into their backs, and they become green. Once they have enough algae or enough chloroplast, they stop eating. And all they do is they just kind of swim into the sunlight because they have eye spots. They can tell the difference between light and dark, and they'll swim into the sunlight and just sit there on the rock and photosynthesize. Right? And it's bizarre because it turns out if you take one of these sea slugs before they've grabbed enough algae and you look at their genetics, their genome, they actually have some genes in the nuclei of their cells that make chloroplast proteins. So somehow they've evolved or confiscated the genes for making some of the chloroplast proteins, and they pass it on genetically. Right? I mean, I think that's cool. But yeah, photosynthetic sea slugs. Okay, go back to the presentation. Okay, autotrophs, as you guys all know, produce their own energy. It's like the sea slug kind of does both. Come on, present. There you go. And they're converting the energy of sunlight to build organic molecules from basically carbohydrates from water. What are the autotrophs going to do with that organic molecule? Why are they building sugars? Okay, what aspect of metabolism? I guess is what I'm asking. Yeah, right? This is something that people tend to forget. I don't know why grade 12 students do this, but they tend to think that animals do cellular respiration and plants only do photosynthesis. Remember that plants do both. They have mitochondria and chloroplast. They do photosynthesis so that they can supply their mitochondria with the sugar it needs in order to do cellular respiration. Okay? Do plants go anaerobic? Do you think a plant would ever do anaerobic respiration? No, they don't, because they don't ever run out of O2, right? It's everywhere. Okay. So they make energy and synthesize sugar through photosynthesis. Okay, so these are the consumers and these are the producers. And you guys know those terms probably from grade 9 ecology. Okay, and we have then these organic molecules cycling around and around between these two major groups. Effectively, cellular respiration and photosynthesis are reverse processes. They're not perfectly reversible, but in general, they're considered reversible reactions. So how are they connected? Sorry that my line's a little weird. So glucose and oxygen gives carbon dioxide, water, and energy, and autotrophs. So you can basically see heterotrophs make energy in organic molecules by ingesting organic molecules, right? So the carbohydrate becomes CO2, okay? It's oxidation, so it's an exergonic process. We then take that CO2, dump it in the air. Plants grab that, and they make energy and organic molecules using sunlight, right? So if we oxidize carbohydrates, no, um, mitochondria came in first, and then chloroplasts were endosymbiosed after. So animals came into existence, animals, big quotes, came into cell existence before plants did. Yeah, mitochondria was actually a response to, so if you mean photosynthesis as the way I'm talking about it, the way plants do it, no. Mitochondria came first. If you're talking about just the light reaction and generation of O2, which is the first half of photosynthesis, cyanobacteria were doing that as an energy source before um, mitochondria came into existence, right? So the cyanobacteria upped oxygen levels 2.7 billion years ago on this planet. That high oxygen level, that increasing O2 level in the atmosphere would have become toxic to all the anaerobic life forms on the planet, and it would have caused a great extinction on this planet. Life was nearly wiped out by the great oxygen event. And then mitochondria and endosymbiosis of mitochondria would have resulted in some proto-eukaryotic cells surviving that oxidation event. And then later, some of the cyanobacteria getting incorporated as chloroplast into that same cell, and then developing the rest, the evolution of the sugar component of the synthesis part of photosynthesis. So the order is a little bit wonky, right? If I, let me see if I can find it. Go to back to grade 11. So courses, no, 3U. Come on, 3U, open up. I don't know why my system is not opening. Am I still online? Yeah. Sorry about doing, yeah, basically. That's right. No one was making sugar per se at the time. I wonder if that's a problem. I've had this issue before. Courses, 3U, evolution. Origin, not kind of here, I think. I think my battery's running out, or my mouse is running out of battery because it's not wanting to open properly. Okay, well, never mind. Anyways, we'll get back. We'll get back to the lecture. Okay. Um. So right, autotrophs are going to make this using CO2 and light energy um, to make the carbohydrate. Okay. Now, yeah, my battery is dead. Okay, hang on. I need to change batteries on my mouse. I'll be right back. Hopefully that's a little better. Okay. Yes. Okay. Now, so being a plant means you need to grab light energy, obviously, and make it chemical, and then you need to store it. So obviously, the most convenient form to store it as in a plant will be glucose. Okay, and we store it so that we can move it around and use it as needed. Okay. You also need to get the building block atoms from the environment, right? So the elements, right? Because a plant cannot make elements. So the elements that it needs, it gets from the environment: carbon, hydrogen, oxygen, nitrogen, phosphorus, potassium, sulfur, magnesium, etc. Right? See carbon dioxide in the air, water from the rain, and dissolved ions in the soil, right? N, P, K are the three big ones. Okay, all of that to produce all the organic molecules that they need for growth, which are these ones that then we need in order to survive. Okay, so in a plant, these elements are the essential nutrients. Okay, in us, it was some of the amino acids and some of the lipids, right? We had essential fatty acids and we had essential amino acids. We have to get it from our diet. For plants, they don't. They can make all of these. What they have to get, what's essential for them, are the actual basic elements. Okay. Now, so just general plant structure. Okay. So obtaining raw materials, sunlight. We're going to do that through the leaves, which are basically solar collectors. Okay. Carbon dioxide is going to come in via the stomates, right? Those little pores on the underside of leaves, and that's via gas exchange. Okay. Water you're going to absorb through the roots. Okay. And then the nutrients you're going to absorb through the soils and the roots. Okay. So the major nutrient collectors in a plant are the leaves and the roots. Now, this diagram or others like it on the structure of the leaf is something I want you to know. I'm not going to ask you to label, well, I might put this on a multiple choice question on a test or something, but I'm not going to ask you to label or draw it. But I will be referring to things like the palisade layer, the spongy mesophyll, xylem and phloem and vascular bundle. You have to know what parts of the leaf I'm talking about when I mention those components in relation to photosynthesis. So you should know the structure of a leaf. Okay. So take a look at this or something similar so that you'll know what and know what the functions of all these components are when we start comparing the different types of photosynthesis that we have. Okay. All right. Who remembers what transpiration is? We would have talked about it last year. Some of you talked about it in the osmosis paper, which I'm currently marking. Okay. Yeah. Basically, evaporation of water going into air. Okay. Which vascular system did this involve? Yeah. So this is part of the xylem transport system. Okay. So the relationship between transpiration and gas exchange is fundamental to actually photosynthesis. So if you don't remember this, please review it. Okay, because this actually leads to some problems in photosynthesis that we need to discuss as well. And we'll be doing that towards the end. So that'll be probably Tuesday, Wednesday, probably Wednesday. Okay. Okay. All right, so let's take a look at the chloroplast, okay, which are the organelles of photosynthesis, okay? They're found in the leaves for the most part, okay? So if you do a cross-section of the leaf and you do a close-up of a single cell, what you'll notice here are all these green little circles. Those are all chloroplast, okay? And what's interesting about this particular shot, if you hit it with a flash of light, those chlorophyll molecules, those chloroplast organelles, will actually migrate and move to where the light flash is. Okay, we'll talk about that. The stringy things in between what? In between the chloroplast or in between the or inside the chloroplast vedant. Okay, so if you look inside the chloroplast, okay, you've got these disc-like structures, okay, and these thin membranes between them. Okay, so that's the internal membrane of a chloroplast. Okay, and we'll talk about that in a minute. Okay, the chloroplast themselves contain the chlorophyll, which is the pigment for photosynthesis. Okay, so you're going to take sunlight and carbon dioxide. Okay, and let me go back. You're going to combine, or you're going to strip the energy from the sunlight and use that to combine the CO2. Okay, how do we do that? Okay, so let's look at the structure of a chloroplast. The chloroplast, again, is a double membrane organelle, just like the mitochondria. Okay, so the fluid-filled interior of the chloroplast, so like this area here, is what we call the stroma. Okay, um, remember that there is an R here. It is not the stoma. The stoma are the pores on the underside of leaves. I have to be picky on this one. If you call the stoma, I have to mark it wrong. So watch the spelling. Okay? So this is the fluid-filled interior. You then have these inner membrane. This inner membrane are called the thylakoid sacs. Okay? And what the thylakoid sacs are is the location where you're going to have photosynthesis occurring. Okay. Now, these thylakoid sacs are often arranged in these stacks. Okay. So a stack of thylakoid are called grana. Okay. And grana is Latin, and it's Latin for a stack of coins. And because they look like a stack of coins, they were called a stack of coins. So that's the grana. Okay. The thylakoid membrane itself, and this thing up here is supposed to represent a thylakoid membrane. My red thing is in the wrong place. It contains chlorophyll molecules. So the chlorophyll is going to be embedded in the membrane. There is an electron transport chain, and there's also ATP synthase. Okay. So what's going to happen? Oh, there it is. So this is a cell. Sorry. This is the chloroplast. There's ATP synthase. What do you notice exists between the interior of the thylakoid sac and the stroma? Hydrogen ion gradient. And since that's ATP synthase, what do you think we're going to use that hydrogen ion gradient to do? Yeah. So this is another chemiosmosis system, exactly or very similar to the chemiosmosis system that we have in mitochondria. So the thylakoid is going to basically have a concentration gradient of hydrogens from the inside out, and the chloroplast is going to use that gradient to make ATP. Okay? That's effectively where we're going towards. Okay, before we can do that, we need to look at light. Okay? What is light? So the visible spectrum, what is it? What is it part of? What do we call this whole thing? Yeah, it's the electromagnetic spectrum. Okay? So it goes from the highest or energy molecules or wavelengths, which are the gamma rays, to the longest and lowest energy, which are radio waves. The visible spectrum makes up a very, very small portion of it. Okay. The part that we're interested in is from, sorry, I'm getting a phone call. Take a bit of a break for now, guys. While they set up my appointment. All right, I'm back. They're gonna call me back again because they're trying to figure out what my second appointment is. All right. So visible spectrum. Um, so we go from high energy to low energy. High energy is shorter wavelengths. Shorter wavelengths means bluer. Okay. Longer energy, lower energy means longer wavelength. Longer wavelength means redder. Okay. So the colors of the rainbow. Why? Come on. There we go. Can you hear me now? Okay. So the visible spectrum, okay, is made up of these colors. Okay. The shorter wavelength, right? Wavelength being the distance between two peaks, is higher energy and is bluer in color. The longer wavelength, obviously more distance between the peaks, is lower energy and is redder in color. These are the colors of the visible spectrum. The only one that's incorrect is indigo. Okay, there are only six colors in a rainbow: red, orange, yellow, green, blue, and violet. Okay, um, so anyone who says there are seven colors in a rainbow is actually wrong. Anyone know why there are seven colors in a rainbow? No, close. It had to do with Isaac Newton. Okay, and Newton was a fairly religious man, right? He actually trained for the priesthood, and he didn't personally love seven, but what Isaac did was he thought seven was a perfect number, and God is perfect. And so when he saw six, he said that can't be right. There has to be seven. So he added indigo as a color. So yeah, that's why we have seven colors today, right? It's the same reason, like in Copernicus's model of the solar system. He thought before Kepler came along, right? Copernicus was convinced that the orbits of the planets were perfectly circular, right? Because they were part of conics in space. So yeah, you had cubes and triangles and spheres that was the pattern that because God would make everything perfect. He wouldn't do something like a rectangle or something weird like that because he wanted everything because God was perfect, right? And those were the perfect shapes. So yeah, religion comes a lot into early scientific findings and has influenced it quite a bit. Okay, so this is the visible spectrum. The only part of the spectrum that we use is this part right here in terms of photosynthesis, right? We can't use ultraviolet, we can't use X-rays, we can't use gamma rays, certainly not, right? That would cause too many, these two would cause too much mutations. Ultraviolet is too high energy, at least with regard to photosynthesis. Plants use UV light in other ways, but not for photosynthesis. Infrared, microwave, and radio, too low energy, they're not useful. So this is the only region that's actually useful for uh, photosynthesis. Okay, so the pigments of photosynthesis. Okay, Everyone talks about chlorophyll as the pigment of photosynthesis. While that is true, it's not the only one. Okay. So chlorophyll is the main pigment, but it's not the only pigment for photosynthesis to work. Okay. So they're embedded in the thylakoid membrane. In other words, there is a phospholipid bilayer in these thylakoid membranes, and the chlorophyll molecule is actually stuck or embedded into that. Okay. We'll talk about the photosystem in a minute. If you look at this molecule, right, this is what photosynthesis looks like. Okay. Why do you think there's a great big long hydrocarbon tail found on the chlorophyll molecule? What will be the purpose of that tail? Anchor it where, Vedant? Yeah. So to make sure that this molecule stays in the phospholipid bilayer, you're going to make this region really, really hydrophobic. That means it's going to want to insert into the fatty acid portion of the phospholipid, and therefore it's going to remain in the membrane. It's not going to float around all over the place. Okay. But up here, this is what we call the porphyrin ring. The porphyrin ring is this structure here with the ion in the middle. In photosynthesis and chlorophyll, it's magnesium is the ion. Okay, you've seen this molecule before. This porphyrin ring also exists in hemoglobin. And in hemoglobin, what do we replace the magnesium with? What's the ion present in? Yeah, iron, right? It's effectively the same ring structure. We just change the ion that's present in the middle. Okay, so this is the portion of the molecule that converts light energy into chemical. Okay, now this chlorophyll molecule is arranged into what's called a photosystem. Okay, so a photosystem is basically an arrangement or a collection of different photosynthetic molecules. All these little green circles represent different types of photosynthetic pigments. Okay. And so they can all capture the energy of sunlight. Okay. So there is a structural functional relationship here. I want to talk a little bit more about the photosystems. Okay. Now, before we talk about the photosystems, let's take a look at how chlorophyll itself works. Okay. So what you're seeing here, yeah, for now it is. That's why I mean, what color are plants in general? I don't see too many red plants. Yeah, most plants are green. Okay. So, and this is why are plants green then? Why, when you look at a tree, well, why, when you look at a green plant, do you see green? Yeah, right? The pigments reflect green light. So, what I want to talk about now is the actual pigments. Okay. So, photosynthesis gets energy by absorbing different wavelengths of light. Okay. There are different kinds of photosynthetic pigments. Chlorophyll A, chlorophyll B, carotenoids, there are others. Okay. So chlorophyll A is here. You can see this graph is a graph of what colors of light these different photosynthetic pigments absorb the best. Okay. What you'll notice is that with chlorophyll A, it absorbs in the blues really, really well. It doesn't absorb the greens that well at all, and it really absorbs really well in the reds. If you look at chlorophyll, so absorbs best in the red and the blue wavelengths and absorbs least in the green. Okay. If you look at chlorophyll B or the carotenoids, right, chlorophyll B, again, absorbs well in the blues, well in the reds, and reflects a lot of green. Okay, but the carotenoids, okay, absorb decently into the greens, okay, but doesn't really absorb in the reds. And then there are other pigments, okay. So what's a pigment? When, so yeah, what is a pigment? We have pigments in paint, we have pigments in plant, we have pigments in clothing. What is a pigment? Right. So what's it mean if you have a white shirt? What's it reflecting? Right. And if you have a blue shirt, what's being absorbed and what's being reflected? Right. Okay. So that's what's going on with chlorophyll as well. Chlorophyll is absorbing everything but green. Okay. And because of that, it's taking the energy from red and blue light and using that to make sugars later. Okay. Carotenoids are what we call an accessory pigment. It doesn't absorb blues or purples very well or reds very well, but it absorbs fairly well in the greens. Okay, so it's a photosynthetic pigment that isn't green. So photosynthetic pigments that are not green are called accessory pigments. Okay, because they assist. Okay, why do you think we have accessory pigments? And we've got a lot of them. Yeah, look at the amount of light that chlorophyll ignores. Okay, so by having accessory pigments that can absorb these colors, we actually get some of the energy in the middle of the visible spectrum to be used by photosynthesis. Okay, based on its name, what color do you think carotenoids reflect? Yeah, right, carrots. Exactly. Yeah, I mean, in theory, plants could be pure black, but and there's an interesting idea as to why they're not, and that has to do with the energy derived. It turns out, actually, I'll talk about it later, but let me see if I can find the UC Riverside paper. Okay, that's the paper I want, but actually that'll load. Okay, we'll take a look at that later. Okay, so going back to this. So these are all different kinds of photosynthetic pigments. Yeah, that's right, Kirsten. Okay. So why are plants green? Plants are green. The easy answer is that plants are green because they reflect green light. But then the question that becomes a little bit more interesting is, I mean, look at the amount of energy that they're missing. Why the hell are they ignoring all of this energy? Okay. And actually, it turns out there's a pretty good reason for it. Do I want to give you that? So there's that one. So I'm trying the article I found. I'm trying to find one that's a little less technical, so that you guys might be able to understand it more easily. Let's see, UC Riverside photoelectric cell news release. Science Daily might be a little better. No, okay. Well, it turns out the reason that plants might actually be green has to actually do with stability. But it actually, well, let me put it up here. Here. Okay. So this is Wired magazine. Okay. And what this person, Nathaniel Gabor, was working on, he's a physicist. He was trying to figure out um, the best conditions for a solar cell, a photoelectric cell, to regulate energy flow. And in the original paper that I read, probably about 5 years ago, what he found was that if you only have one solar cell, the intensity of sunlight, right, the amount of light that falls on that solar cell is going to vary. And as the amount of sunlight varies on the solar cell, what's going to happen to the amount of energy that's going to be pumped out by amount of electricity that the cell can pump out. So if sunlight intensity goes down, what's going to happen to the energy output of the solar cell? Right. And then if it gets sunny again because the clouds go away, the energy output's going to go up. And so the stability or the fluctuation, now that kind of fluctuation on the power grid is really, really hard on electrical equipment, right? What you really want is a constant or relatively constant energy source. You want electricity levels to be pretty constant so that you can shoot at long distances. That's why we use AC current, not DC in North America. If you're in France, they use DC. If you're here in North America, they use AC. Why? It's more efficient. It's more stable. You can send at longer distances with fewer transformers. Okay. Now, in plant, it turns out when he was doing this solar cell, if he wanted to get two solar cells, if he wanted to get electrical stability in terms of energy output between his from his solar cells, what he literally had to do was he had to set up two solar cells in tandem with each other. And they had to collect sunlight at two specific wavelengths. What color of wavelengths do you think it was that gave him the most efficient stability of power? Yeah, well, more blue and red. So he was using about 450 nanometers and 630, 640 nanometers wavelength, which are coincidentally looking right here, the same two wavelengths of light that plants use in order to collect sunlight for photosynthesis. This was a bit of an aha moment for him. The first paper I saw was just a real small news release. This paper now, or this Wired article, actually goes into it in detail about how this works. Um, my question, I mean, we don't have time to look at it right now. We can maybe take a look at it later when we've done the light absorption stuff. But in the meantime, do you want me to post this article? Are you interested enough in it that I should post this article in Classroom? Yes? Okay. So you may not understand it right yet because we haven't completed all of the light reactions yet. But certainly, once we finish the light reactions, I think you should be able to understand the article. So I'll put it in with the light reaction stuff. So I'll add it there. Edit, add a link, Control V, add link, save. Okay, so it's on, it's just called Why Are Plants Green, and it's at wired.com. Okay. In part, Graham. Sorry, Grant. Um, they're green initially, not because of photosynthesis. They're green because the starch inside the fruit is still starch, right? And what are fruits used for? Well, that's the ultimate result. But yeah, how are they used in spreading? Get animals to eat it. Now, if the seed is still starchy, it's still starchy because the embryo is still developing inside the seed. If a plant eats an developing embryo, the likelihood that that embryo will survive is pretty low, and it'll be digested. So what plants do is they keep the starch inside the fruit starchy until the embryo in the seed is developed. Once it's developed, they're going to use other chemicals, ethylene gas primarily, to ripen the fruit. And the starch gets converted to glucose. It sweetens it. That sweetness attracts plants or herbivorous animals or omnivorous, gorillas, whatever, to eat it. Okay. And plants signal that they're ripe to eat by coloring the red, yellow, orange, purple, black colors. And this is why a lot of primates and a few other, the herbivores that eat fruit, all developed color vision to be able to tell the difference between ripe and unripe fruit, in part, is one of the theory. For example, it's why dogs don't see in color, right? What do dogs normally eat? Well, meat, right? So because dogs usually eat meat, right, their visual receptors are set for orange. So they're not black and white. They see sort of shades of orange all over the place, right? That's what dogs basically see. But your primates, anything that can detect color vision, right, is going to usually have the is doing it for one of two reasons. It's a long distance hunter, things like raptors, right? 4 kilometers up can spot a rabbit. They need to have some color dexterity there. Or you're talking about some animal that's going to eat ripened fruit. primarily, right? Horses, giraffes, elephants don't have huge color vision because they don't really distinguish fruit from grass because they can digest grass. They're herbivores, right? But anything that focuses mainly on fruit, they're going to have color vision or something similar to it. A lot of insects see in the ultraviolet because flowers actually have pigments that reflect ultraviolet light and give them targets to look at. We'll talk about that later. It's 10 o'clock. Shall we take a bit of a break and then come back in about 10 minutes? Okay, and we'll continue this. So we'll see you in 10. Okay, and we're back. It's a little bit over, but anyways, let's keep going. All right, so this is the action spectra. So this previous image is an absorption spectra. So it shows you what colors of light these pigments absorb the most. The higher the peak, the more color it's absorbing. Okay. The next graph is a light action spectra. What this graph tells you is the relative amount of photosynthesis that a plant does under different colors of light. Okay. So it shows the relative effectiveness of different wavelengths of light on allowing photosynthesis to occur. So what do you notice about the peaks of the action spectra compared to the peaks of the absorption spectrum? They're effectively the same, right? They peak at the same points, blues and reds. Okay, basically this. This is the evidence that early uh, botanists used to collaborate, right? That photosynthesis was the primary pigment for plants use, right? Because they matched up so well. It wasn't necessarily chlorophyll. It wasn't necessarily causation, but it correlated really, really strongly. Okay. So, and before we talk about this, how do we know that there are other colors of light that, or the other pigments that also are involved in photosynthesis? How do we find them? Or how do we at least suspect they might be there? Right. But what's the evidence for it? Before this, people had a suspicion. When do we see other pigments of photosynthesis in a plant? Yeah, in the fall, right? What colors do trees go? What are the major colors that you see in a deciduous tree during autumn? Yeah, those are the three biggies. Okay. Why do you think those colors appear at that time? What changes in a plant, in a deciduous plant, that we don't see the green anymore? Why does the green disappear? Yeah, but why? No, not because of no need. In part, in part, but I mean, they could, right, just drop the green leaves. Okay. Yeah, the question is, why is the plant taking the chlorophyll away? Yes, less sunlight is the stimulus for this to occur. Yes, that's absolutely true. Because with the higher angle of the sun or the lower angle of the sun, the other colors become more effective at photosynthesis. That's true. But why not just drop green leaves? Why remove the chlorophyll from the leaves? That's what's literally happening. It's disappearing. Close, Kirsten. Chlorophyll requires a lot of energy to create. So Vedant's closer. The plants absorb that chlorophyll, right? Reabsorb it, and then they're going to reuse it later in spring. They're going to put it back out so that the new plants, the new leaves, can actually use it. And so because the chlorophyll value, amounts start to decline, we see the other colors of photosynthesis. What's interesting is that we don't see that in conifers, right? What colors do conifers go? Yeah, that's why they're called evergreens, right? Think of your Christmas tree, right? The Scotch pine is a green plant year round. in part because of the evolution of leaves, rather of needles for leaves rather than your standard broadleaf, right? Is part of the reason, not the only reason, but part of it. Okay, let's take a look at these photosystems. Okay, so so imagine all the white area is actually the phospholipid bilayer, the phospholipid heads. Okay, the light green discs are all the other pigments of photosynthesis that can absorb the different colors of light, and the dark green is actually chlorophyll. Okay, a specific type of chlorophyll. Okay, now, so this collection of pigments is what we call a photosystem. Okay, there are two photosystems in the chloroplast membrane. Okay, and these are basically collections of chlorophyll and other accessory pigments. Now, there are light gathering molecules, okay, that are going to collect the energy of sunlight and use it to drive photosynthesis. Okay, the first one we are looking at is called photosystem 2. It's called photosystem 2 because it was the second one discovered, but it turns out it's the first one used by plants. Okay, it's the first one in the sequence. Okay, so the main chlorophyll molecule, the one that we call the reaction center chlorophyll, is chlorophyll A, the dark green one. Okay. It's also known as chlorophyll P680 because that particular chlorophyll molecule specifically absorbs wavelength of light at 680 nanometers. In other words, it absorbs red light, that particular molecule. So if red light at that wavelength hits it, this molecule is going to react. But what if light hits it at a different wavelength? Okay. And I'll talk about that in a second. The other photosystem is photosystem 1. Okay. It uses a different chlorophyll molecule at the reaction center, chlorophyll B. It's called P700 because it absorbs light at 700 nanometers. They're both absorbing red light. Photosystem 2 absorbs 680. Photosystem 1 absorbs 700 nanometers right here. Okay. The rest of these pigments are what we call antenna pigments. So they're the other pigments of photosynthesis. Could be xanthophylls, anthocyanins, carotenoids, it doesn't really matter. Okay, so what are the antenna pigments for? Well, if a photon of light comes in and hits it at a wavelength other than 680 or 700, one of these other pigments are going to capture that energy. The energy then is passed on from one pigment to another until sufficient energy is absorbed that the reaction center chlorophyll can use that energy to make photosynthesis work. Okay. How this work is way beyond the scope of this course. It involves something called quantum vortex tunneling. It is something like a third year quantum physics problem, and it's not something I'm going to deal with. Okay. But it has to do with the state of electrons between two different antenna pigments and how that electron moves and shifts from one molecule to another. Way above and way beyond the math and the integral, partial differential, and integral calculus you need to understand what's going on, well beyond the scope of this course. Even if you have calculus, you only have full integral and full differential. You don't have partial calculus yet, so there's no way you could work this out. You're going to have to wait until you have more physics and math, okay, if you're interested in this. Okay, I mean, that's what they were doing when they were working on this paper, right? The why this thing is using that level of mathematics. Okay, so just understand that if energy, yes, until the reaction center chlorophyll. So you just need to understand that if wavelengths of light other than these two are absorbed, then the antenna pigments will deal with it and pass that energy on to the reaction center chlorophyll. The how is not important for grade 12 biology. You just understand that's what literally happens. Okay? So this is what we call the reaction center. This is where the main business of photosynthesis, or the main conversion of light energy to chemical energy, is basically going to occur. Okay, when we talk about this conversion of light to chemical, how is it actually done? All right, we're actually going to talk about a process called fluorescence. Now, if I, so photosynthetic fluorescence, let me see if I can find a, okay, this is usually, now, what you're normally used to seeing when you see light hit a plant is green light reflected to you. Okay, that's what you're used to seeing. But what do you think would happen if you took just the chlorophyll? So let's say that you took spinach leaves, because I've done this in class before, right? You put it in some alcohol or some acetone in a mortar and pestle, and you crush the leaf so that you get a solution of chloroplast or chlorophyll. Usually it's just a solution of chlorophyll because you grind the leaf up. So you get a solution of chlorophyll suspended in a solution of either alcohol or acetone. And then you shine light on it. What do you think would happen? Okay, let me show you what happens. Right, but what is fluorescence? What color would you see? Okay, so here's what you get. This is the image here. So this is a solution of chlorophyll, okay? And if you hit it with white light, this is what you're going to see. It fluoresces red, okay? And this is what we call fluorescence, okay? So if you take any chlorophyll molecule and you hit it with a high energy photon, you will see energy reflected out of that photon, and you'll see a lower energy light shown. No, it's not the opposite. Okay, so here's what's going on. Okay, now instead of using a compound like chlorophyll, I'm going to simplify it by using a single atom instead. Okay, so here in light blue is the nucleus of an atom. There's an electron sitting in its orbit. Okay, and what we're going to have for fluorescence to work is you're going to have sunlight come along. Okay, and that sunlight photon is going to have the perfect wavelength that when it hits this electron, that electron is going to absorb that energy because it's the right wavelength. Okay? Because it absorbs that energy, what's going to happen to that electron? Right. So it's at ground state right now. The electron is going to jump up to a higher energy state. Okay? Can the electron remain at that high energy state? No. It has to fall back down. So when it falls back down, that energy has to be released. The energy that's released is a lower wavelength of light that gets emitted. And that's what we're seeing here. High energy light came in, the chlorophyll molecules, basically the electrons in the chlorophyll molecule, jumped up to a higher energy state. They then fell back down, and that energy was released as glowing red light that we can see here. Okay, so that's what chlorophyll does. Effectively, yes. If this was a metal, we would call it the photoelectric effect, but it's a plant, so it's not. Okay. Now, so it's equivalent to the emission spectra that you see, yeah. In metals, yes, not fluorescence in plants. The photoelectric effect is what gave him the Nobel Prize, yeah, in metals. Okay, so these are all the different, or some of the different photosynthetic pigments that you can get out of a plant using chromatography. You can see the different colors that they reflect. Okay, so paleophyton is more of a blue dark color. Chlorophyll A, chlorophyll B, lutein, vitaxanthin, neoxanthin, right? All the different colors. They are all going to do this. Now, the question is, how does a plant use this? Because it uses that effect to convert light energy to chemical. And the question is, how does it do it? Okay, let's go back to here. All right, the process is no longer fluorescence. The process is now photooxidation. This is what a plant does. In an intact chlorophyll, so in the previous animation, that was just chlorophyll molecules on their own. And so the electron would jump up, jump down, and emit light. Okay. This time, we're in an intact chloroplast. So all the components for photosynthesis are there. Okay. So now what happens? Well, we're going to start the same way. You're going to have sunlight come in. You're going to have a photon of light with the appropriate amount of energy come in and hit that electron. That electron will absorb that energy and jump up to a higher energy state. So far, exactly the same. Here's what changes. Now, because we're in a complete chloroplast, and the electron is now at a higher energy level, it's easier to remove from the molecule than it would be if the electron was at its ground state. So what can actually happen is NADP plus will come along and grab the electron and become NADPH. Okay, so the electron was removed or transferred from the chlorophyll molecule to the NADP plus molecule. NADP plus was reduced to NADPH, and the chlorophyll molecule is oxidized. Okay? Because the oxidation occurred due to the absorption of light, this process is called photooxidation. Key point. Plants use NADP for the electron carriers. Mitochondria use NAD for the electron carriers. Don't get those two mixed up. You do. I have to mark it wrong. Right? P for plants. So plants use NADP plus going to NADPH. Mitochondria use NAD plus going to NADH. Okay? So that's how plants convert light energy to electric, or light energy to chemical. They basically steal the electron when the electron jumps up to a higher energy state, and now that's captured as chemical energy. Okay? All right. This is where, let's see, how it's 20 minutes. I don't know if I'll be able to cover this in 20 minutes. I would normally do this on a Blackboard, but obviously we're going to do it on a Jamboard. So let me start that up. Here we go. Open a jam, start a new Jamboard, send, show this tab. Okay, maximize. All right, I cannot see the meat now because I have the Jamboard filling my entire screen instead, and I don't think I can move you guys over to the Jamboard. I don't think I can pull you out and add you to the jam. Let's see, can I? No, I can't. All right. So I'm going to minimize my meet. I won't be able to see any questions if you have any questions in the chat. So if you have a question, you're going to have to unmute. Okay. So people are on. I am sharing the correct tab. Okay. I'm going to build my. So if you look at this, right, the slide deck slide that I've got here, this is actually a graph. Okay. And it's called a Z graph because of the shape of the pathways, right, is a zigzag. So it's what we call a Z graph. On the vertical axis, the energy level of the electrons. On the horizontal axis is basically where you are in the photosynthesis, in the light reactions. I'm going to basically build this diagram in the Jamboard. Okay. So the first thing I need are axes. So that, there's my two axes. Okay. And my vertical axis is the energy of the electrons. Yay, I can move it. Okay, so the energy of electrons is my vertical axis. And my horizontal axis, I'm just going to call the light reactions. Okay. All right. I'm not going to draw the entire photo system. I'm just going to deal with P680 and P6 and P700. Okay. I'm going to start with photosystem 2. Right. You can see here in this diagram, right, the process starts with photosystem 2 and then goes on to photosystem 1, which is why I'm also starting with photosystem 2. Okay. So this is P680. Okay. So that's my chlorophyll molecule in ground state. Okay. Light hasn't hit it yet. It's just sitting there in my leaf, minding its own business. Okay. Now I'm going to have light come along and hit it. So I'm going to have yellow light come in. And I'll have the light coming in from the bottom, I think, because it'll be easier that way. So that squiggly line represents sunlight hitting P680. Okay. Since it's light at 680 nanometers wavelength, it's going to hit the appropriate electron, specifically two electrons, and two electrons are going to get pumped up to a higher energy level. Okay. So I'm going to have two electrons that are basically going to jump up. Oops, to a higher energy level. Okay, so there's going to be two electrons that are moved up. Okay, when that happens, Okay. The name I'm going to give to this molecule, oops, I don't want that duplicated. Can I? This molecule is now designated P 80 star. The star indicates that this molecule is, or the electrons in this molecule are now in a higher energy state. Okay. What I said before was then that the electrons get ripped off. Okay. Photooxidation occurs, and this is true. So it goes from P680 star to a molecule here. I'll make it big enough that I can type in it. And that molecule, this electron molecule, is a molecule called PQ. Okay. And PQ, put it up here, I guess, is a molecule called, oops, plastoquinone, not quinine. Quin is a molecule called plastoquinone. So the electrons are literally gonna, the two electrons are literally gonna move from here. Oops, erase all that. Keep forgetting to hit my shift key. Shift, there we go. So the electrons are gonna move from there to there. Okay. Then they're gonna move to something. Let me make it big enough. Called a cytochrome complex. Okay. And literally, the name is cytochrome complex BF6. So the electrons are going to move there. However, time, 10 more minutes. Okay. Now, I'll talk about this specifically when we do the actual model of how this thing works. But cytochrome BF6 is the point where we're actually going to have ATP being made. So we're going to, at this point here, I'm going to put in another line that goes like this. Oh, so this is where we have ATP synthesis. Okay. Not going to talk about how yet. Just know that this is a complex where ATP synthesis is being done. Okay. We then go to another molecule about here, which is called PC. And PC is a molecule called plastocyanin. Okay, so again, we're going to have the electrons passed on to there. Finally, we're going to have the electrons will finally pass back to the second photosystem. So this will be P700. Okay. So what we've done is we've sent electrons from P680 through an electron transport chain. Okay, that's what this is. So I should probably put that there. This is what we call an electron transport chain. Okay. Down and in the process of using this electron transport chain, we're going to use chemiosmosis to make ATP. And in the end, we give those two electrons that were initially in the P680, they end up in P700. Okay. Now that we're in P700, let me just go back to the meet to make sure that you guys are following this. Okay. Any questions up to this point? I'm looking at the meet now. Up to this point, is there anything that's a little confusing and you need me to go over again? Hands raised if you do. No, just an exercise program. They're not in ATP synthesis, just like the electrons in they're used to power ATP synthesis, just like in the mitochondria. It's chemiosmosis, right? It's a hydrogen ion gradient. You don't. The electrons never end up in ATP itself. Remember, in the mitochondria, you have the electrons going from NADH to oxygen. That creates a hydrogen gradient, which is used to make ATP. Same thing here. You're going to have the electrons moving from P680 to P700. That's going to create a hydrogen ion gradient, which is going to be used to make ATP. So the electrons never end up in ATP. Okay, so now this is also a photosystem though, remember? So I can have another wavelength of light come in and hit it. This time the wavelength has to be 700, okay? So 700 wavelength light hits, and the same thing happens. I'm going to give myself a bit room by shifting the arrow over a bit. And you're going to have the fact that I drew them at slightly different height doesn't mean anything. So this is now P700. I'm going to shift it over so I give myself a bit more room. The horizontal positioning of the components doesn't really matter. I'm just running out of room, so I'm just moving things around a bit. So obviously the energized state is P700 star. Whoops, no and. Okay. So we've again elevated two electrons to a higher energy state. Okay. What we then have is something called FD. And this is actually another ETC, another electron transport chain. Okay, and FD will Ferrodoxin is the name of this compound. Okay, so you're going to have the electrons get past. Oops, I did it again. Damn it. So the electrons get passed from P700 to ferrodoxin. Okay, and then finally, the last thing that takes it is there's going to be another molecule, and I may not have enough room for it. We'll see. Which is called NADP plus reductase. That's the name of this molecule. And so the electrons, again, are going to get passed from there to here. What do you think NADP plus reductase does from its name? Reduces the NADP plus. So what's going to happen is that you're going to have NADP plus is going to basically come through here, pick up the 2 electrons, pick up 2 hydrogens, and become ADPH. Oh, the H fell off. Sorry. Okay, so it'll become NADPH. Okay. So this is also an electron transport chain, even though it's a much shorter one, right? And this ETC does not produce ATP, but there are 2 electron transport chains in photosynthesis. Okay. All right. Almost done. Looking at this diagram, okay, there is one significant problem for one of the chlorophyll molecules. What is it? All right, I'm coming back to the meat, so give me a sec. Okay. So NADP plus reductase is an enzyme, as Abby said, that reduces NADP plus to NADPH. Eventually, one of the 2 photosystems is going to run into a problem. What's the problem? Yeah, P680. And Vedant is exactly right? Because here's what's going on. P700 will always, when it loses its electrons, the electrons will always be replaced by P680. But P680 has no source of electrons right now. So as light keeps hitting it, its electrons are going to keep getting shot through. And look, they end up in NADPH. And what NADPH does is this goes to Calvin cycle. Okay, so the NADP goes off to the Calvin cycle to make photosynthesis occur, to make the sugars. The ATP is also used in the Calvin cycle. So both of the ATP and the NADPH are what we want to make the sugar, right? This is why we do the light reaction. But the problem right now is how do we replace the electrons that get lost from here? What do you think the source of electrons will be a good source of electrons for P680? So where could P680 get electrons? Yeah, it uses water. So what's going to happen is that water is going to get split into two H plus plus a half O2. Okay. So when that happens, so when this goes to this, you're going to basically have two electrons move here. You'll have two electrons move from the water to P680. Once you have two water split, what are you going to release as a waste? That's where the O2 comes from. Okay. Now, there's an enzyme that does this. The enzyme is called a Z protein. I don't think I can fit it in there. But you probably want to make note of that somewhere. That Z protein splits the water. Okay. That would normally go there. I didn't give myself enough room. Okay. So the enzyme effectively that splits the water in two is something called a Z protein. Yes, it will, because eventually they'll get pumped across that membrane and help build the gradient. Okay, so that's the light reaction. That's the complete light reaction, right? And I figured it was better to build it that way than to just give you this all at once. Okay. So do you want this jam available, or are you okay with the graph in the text? Yes, you want it available. Okay, so let me just title it and then put the jam into. So the title is going to be Z diagram light reaction. Okay. I'm going to close this and post it so you can log out if you like. Now I need to find classroom. Okay, so let's edit, add. Is it in Google Drive? Yes, Z diagram. Insert. Save. So if you look at, where are you guys? This, because oxygen is O2, and you only need one oxygen for water. Okay, so there's the Z diagram that we just made. The same diagram as this, 10b. It's sideways, but it's the exact same idea, right? There's a Z protein, water. It does take energy a little bit, but you can ignore that. And then the creation. This isn't one-to-one, which is what it looks like. You're not using one ATP and making one ATP, so net is zero. That's not the way it works, even though this diagram makes it look like that. Okay, so you actually produce more ATP than you actually need for this process to run. Okay, and then this ATP and this NADPH are used in the Calvin cycle to make the sugar. Okay, so that's where we're going to stop for now. On Tuesday, I'll continue with this, but I'm going to show you exactly what happens in chemiosmosis. How's the ATP generated? Then we'll look at the dark reactions. All right. So that's it for now. You guys are dismissed, and I hope you have a good weekend and enjoy the sun a bit. Get outside.